in just a couple of minutes after the hour as people are joining in. But if you are here for a webinar focused on the wonders and opportunities and challenges of mRNA as a platform for HIV vaccines, you're in the right place. And we will get started in just a minute or two. Welcome everybody, though. All right. Welcome, everybody. My name is Mitchell Warren, the Executive Director of AVAC, and delighted to welcome everybody to this webinar related to HIV Vaccine Awareness Day. I realize it's a minute or so after. We are going to get started in just a second. We have what I think is an amazing lineup of presenters, and we want to get started because we really want to get through their presentations in the first half of this conversation so that we can turn to a discussion with all three of them in the latter half of the call. And we will start in just a minute. Well, I think I will go ahead and get us started in the interest of time, and because the least important thing is to hear my introduction, and we want to get into the conversation. Again, welcome everyone to the first of two webinars, and then later in the call we'll do an update and give you some registration details and background for next week's second webinar looking at antibodies and their relationship to HIV vaccine research. Both today and next week are part of our conversations related to HIV Vaccine Awareness Day, a day that's been marked now since 1997 as a time to reflect on and recommit to the urgency to develop an HIV vaccine. I do want to begin before I jump into the content today. Obviously, nothing in HIV vaccine research or in the HIV response really can appropriately be addressed without thinking about the social, political, and structural issues. And it would be impossible to think about a conversation about anything related to HIV this week without calling attention to a heinous decision by the president of Uganda earlier this week in approving an anti-homosexuality act and putting it into law in Uganda. I raise that because whether you are an HIV researcher, an mRNA product developer, an advocate, anyone working in global health has to be aware of and alarmed by not just what's happening in Uganda right now, but what we at AVAC regard as a really global cascade of terrible policies that inhibit our ability to deliver HIV programs and services and research. And so I wanted just to take a moment at the top of this call to acknowledge that and to recognize that even while we will spend the next hour or so talking about vaccine research, that we fundamentally understand that it connects to the much larger issues of human rights. And look forward to working with all of you in Uganda, in Africa, and around the world to ensure that we do all that we can to make communities safe, to make programs exist in safe and legal spaces that also help research, and just want to acknowledge that today. But we are going to focus today on vaccine research and development, and it comes at a remarkable time. In the midst of COVID, we have seen some of the most remarkable advances in vaccine science, and many of us have talked over the last 3 years that little of the COVID vaccine process in research and development and the wonders that occurred over the last 3 years would have been possible without the foundational work of the last 30 years of HIV vaccine research. Whether that's funding, whether that's clinical trial networks, whether that's product platforms, whether that's models of community engagement. None of that could have happened as quickly as it did in 2020 had it not been for the power of HIV vaccine research and development. So everyone on this call and all of us working in this space, I think need to acknowledge that while we still struggle in developing an HIV vaccine, that what has been built by this global community had one of the greatest, was one of the greatest assets in COVID vaccine development. And I think all of us have wondered since that time of rapid vaccine development, what are the lessons for HIV vaccines? And that's really where we want to go today. We have three terrific presenters who are going to give you different perspectives on mRNA as a platform and on larger conversations of local manufacturing of vaccines. Because we know that as great as the science was in COVID vaccine development, the ability to deliver it at scale and most of all with equity around the world was inhibited. And one of those issues certainly was issues of manufacturing and intellectual property. So we're going to look at all of those issues as they've played out and as they play forward for HIV vaccine research and development. We were tempted to do a poll at the beginning of this. How many of you knew about mRNA as a technology before mid-2020? You're welcome to show your hands. There's at least one person who says absolutely. mRNA was not new because of COVID, and in fact, some of the earliest work in mRNA technology was in the context of HIV vaccine research for decades. But it was really with the advent of COVID vaccine development that mRNA really began to find its home in vaccines that were licensed now. And now the question, what, if anything, can be done to accelerate HIV vaccine research? And as many of you know, and you'll hear about it on this call, several vaccine candidates now in early stage development for HIV, as well as for TB and other pathogens using mRNA. So we're going to focus on all of those issues. We're going to have three presentations. They are wonderful and relatively brief. We're not going to stop for questions in between, but I encourage you to put questions in the Q&A that you'll see at the bottom of the Zoom and or in the chat. We'll monitor that, and then we'll come to a live Q&A with all three panelists after they present. And again, we'll feel free to take your hands if you raise them to give you voice as well. So that's the run of show, and delighted to welcome you all here. We're going to start, actually, I think, in a really exciting and interesting place, and that's with Ike James of the Medicines Patent Pool. And many of us working in HIV, particularly over the last several years, have come to see MPP as a critical partner in thinking about product development, product manufacturing, and eventual access. And particularly in the HIV space, the MPP, as many of you will know, has been a leader in accelerating the licensing of injectable cabotegravir for PrEP. Just 2 months ago now, MPP granted 3 licenses to 3 generics. But at the same time, many of you may know or may not know that the MPP has been a key partner in developing what is now the mRNA hub, which we'll hear even more about in our second presentation. But Ike, relatively new to the MPP, is involved in leading the technology transfer program of the MPP, working very closely with WHO. And it's really exciting to welcome Ike. Ike has actually a vast background in generic manufacturing, as you'll hear about, I think, and is really leading the ability to partner in the transfer of technology, including particularly those that are complex in formulation, in terms of building up local manufacturing capacity. So Ike, welcome from Basel, Switzerland, and originally from South Africa, but currently in Basel, to provide our first insight into mRNA and local manufacturing.
Many thanks, Mitchell, for the warm introductions. Really pleased to be able to be here at this webinar sharing with you this presentation. Yes, I started at MPP just over a year ago.
And Ike, before you, hate to interrupt, but if you want to go into presentation mode.
Thank you.
There we go.
All right, thanks. Yes, so I started at the MPP over a year ago, leading the technology transfer team. And we're a handful of technology transfer experts that are really supporting MPP in its efforts to support the mRNA hub, and also support MPP in its effort as it expands into new complex formulations and biologicals. So today I'm really going to focus on our work on the mRNA technology transfer program that we are co-leading with the WHO. And this is basically focusing on catalyzing and accelerating local manufacturing production capabilities to really address and improve equitable access. Following me is Caryn and Mike, and really going to complement this talk. So I will try and be brief because I think you really want to listen to them. And in this presentation, I'll give you basically the overall framework of what we are trying to do in this program. So the agenda that I'll be talking to you today will really be focusing on why is a global effort to improve access equity necessary? What does it take to achieve this? And how do we plan to achieve this through the model of a mRNA technology transfer? So let's start at the beginning. Equitable access. If we take a snapshot in August 2021, we see that 70% of the European adult population was fully vaccinated. And you can see the disparity that existed in Africa, South America, as well as Asia. Focusing, if we fast forward to as recent as May 2023, we see that a lot of the global north, this issue has been addressed, with 30% of people in low and middle income countries having only received at least one dose, with a major focus on Africa at large still lingering behind. So this has really brought this mRNA technology transfer program into focus, where it was initiated in April 2021 with a clear focus of addressing this inequity and looking at improving health security in these low and middle income countries through establishing local and regional production and a focus on mRNA vaccines. So the objectives were to introduce or expand know-how of this mRNA technology in these regions, as well as establish or enhance sustainable mRNA-based vaccine manufacturing capacity in regions with no or limited capacity. And secondary objectives was really to support the local workforce development and strengthen the regulatory capabilities, which we see as key enablers in really enhancing this capacity. In terms of what it takes, there are numerous factors that need to be considered. Technology transfer, the transfer of knowledge, documentation is one thing, but there are numerous other elements that really need to be taken into account. You need to look at the local workforce, what it takes to manufacture vaccines. We need access to state-of-the-art technology and processes. This does not, in the past, we've been looking at egg technology, which is 60-year-old technology, but mRNA is really cutting edge. This is what we have chosen for numerous technical reasons, which I'm sure Caryn and Mike will take us through. But cutting edge technology is really key for our partners. We see that R&D capability and capacity is also an essential ingredient, as well as bricks and mortar, the actual infrastructure where these R&D and manufacturing can be activities can be executed. The regulatory system is key, together with coordinated workforce and opportunities development to ensure that we work in a coherent way without duplication of efforts. Coherent health, economical and industrial policies are key, as I'm sure you are all aware. as well as access to education and specialized training. Last but not least is obviously engagement and commitment, not only on the short term during when a pandemic exists, but long-term commitment is really important. And now that we see that the COVID pandemic has hopefully come to an end, this long-term commitment that takes us and really cements the future pandemic readiness and inter-pandemic sustainability are really key activities that we need to follow through on now. So what have we done? We have established an mRNA technology transfer hub. And this hub its aim is really to establish a technology and validate it. This is in the absence of a technology being made available by Big Pharma that had this know-how already established. So you'll hear from Caryn as to the efforts of what Afrigen and the South African consortium have had to do into establishing a technology platform that can be used as the framework that we can not only technology transfer to our partners, but also allow us to exercise improvements to this technology that can then be shared globally with all of our manufacturing partners. And this also encourages the likes of inventors, experts, developers, IP holders, and researchers all to have input into this hub and to really make integrated R&D and manufacturing network. And we can talk a little bit about that a bit later. So the hub and partners, let's maybe go through a little bit of detail on that. So the mRNA Technology Transfer Hub has been established in South Africa. We have Afrigen who have developed this mRNA technology and established a process for early clinical development. They are also the training center and sending unit for the technology transfer. So you'll hear from Caryn about how the training center is working and how we envisage technology transferring all of the process units as well as all of the analytics and supporting of our manufacturing partners during this process. We have Biovac in Cape Town in South Africa, which will be the first manufacturing partner to receive the technology from Afrigen. And they have a really critical point to play in this, where they will scale up the process and validate it to a commercial scale and validate the methods that is in line with a commercial, that is fit for purpose for commercial needs. A critical component is the further development, further improvement of the technology to next generation technologies. And this is where the South African Medical Research Council are working on. This is obviously to support innovation. We feel that the first generation mRNA technology is the first step, but a continuous improvement of not only the technology, but working on a pipeline of products, including HIV and TB, as well as other disease areas that are relevant to our regions of our manufacturing partners is really critical. Below you've seen we have 15 manufacturing partners that have been nominated through an expression of interest. And these are based around the world. You'll see South America, we have 6 in Africa, we have Asia, we have also the Ukraine and Serbia in Eastern Europe. All of these manufacturing partners are key, and we have engaged with them in training exercises. A month ago, we had, in April, we had a key meeting in Cape Town to really galvanize us as a network. And we're looking at co-development activities at next disease areas, forming CRADAs with them, as well as looking at how we can learn from each other. So this is really a network of manufacturers that is encouraging a network of research and development. This is my last slide, and basically the fundamentals of this model. I think the uniqueness of this model is firstly looking at a tech transfer approach that is not looking at bilaterals, but a multilateral technology transfer approach. We're looking at this manufacturing partners network where we can learn from each other. We can look at integrating this first generation platform, looking at improving that, looking at improving the cost of goods further, looking at improving the thermostability. We all know mRNA in terms of thermostability with -40 to -80 storage conditions brings in complexities. But if we are able to bring this down to 2 to 8 or even room temperature, would be an added bonus. This network would also encourage R&D with the further improvement of this technology for disease areas relevant, such as dengue in Asia or RSV for Africa and Latin America. So there's a continued effort to align on what are the priority disease areas per region. And I think that the message that we want to do, we want to build this model that looks at having this manufacturing and R&D network that also looks at the freedom to operate, where the R&D is focused on generating technologies where we have the freedom to operate and we share this knowledge with each other and technologies as well. So I'll pause right there and I'll let Caryn or hand over to Mitchell for any questions, or handing over to Caryn. Thanks very much.
I thank you. That was terrific and really set the stage for this overall conversation. And it's great to see maps that are driven by the Global South in driving these issues. So thank you for that. As Ike said, a lot of this focus has centered in Cape Town and at Afrigen, a leading South Africa-based biopharmaceutical company. And we're delighted to have Caryn Fenner here, who is based at Afrigen and has been for a number of years, and more recently has been become the executive director of the mRNA hub at Afrigen. And we're delighted, Caryn, to have you here. And it's been exciting to watch over the last couple of years to see the hub go from a concept to actually facilities that are really driving and generating great insights. And so I'm happy to turn the virtual floor over to you.
Thanks very much, Mitchell. Let me go into presenter mode.
Beautiful.
So I just want to apologize if there's bandwidth issues. So we, as you know, in South Africa, we're struggling a little bit with our electricity challenges, and then that sometimes has knock-on effects with our internet. So really want to say thank you to Mitchell and the team for inviting Afrigen to be part of this webinar, and really good to have everybody online. I have a few slides. We have a lot to say, so I'm going to try to go quite quickly, and Ike has actually shared some of the information, so you'll see a little bit of overlap, but I'll try to just go through that quite quickly. So just in terms of me, I head up the mRNA hub. I've been with Afrigen for 7 years, since it became operational in 2016. And I thought I'd start off by just showing you the timeline, Afrigen's timeline, and with that, giving you a very short overview of the history of Afrigen. We started out in 2016, and our original mandate was to localize vaccine manufacturing. So it started with a very short 18-month project off of the back of a TB vaccine, which we in-licensed from a nonprofit organization based in Seattle. Some people may know of them as IDRI. They are now renamed as the Advanced Access to Health Institute. And so this TB vaccine was being tested in clinical trials just at phase 2A for the prevention of recurrence of TB. And with that, we in-licensed a portfolio of proprietary adjuvants, and adjuvants go as a complementary technology to vaccines to deliver the antigen and or to modulate the immune response. And so being a young company, And looking towards commercial viability, we had to think about how we could use our formulation technologies and apply them to other health products, getting them to the market faster. And so we did that with complementary and alternative medicines as well as nutraceuticals. But we continue to build the relationship on the adjuvant side with antigen and vaccine developers. So reaching out extensively to our local network, but also internationally. And started to build a portfolio of vaccine candidates that we wanted to see through to the market. So at that stage, we were able to convince our shareholder that we needed to build a vaccine facility. And the idea there was to see our own vaccine candidates through to the market, to be able to make clinical trial material, because in Africa we have no such facility for human vaccines. And we wanted to also focus on neglected diseases. And so essentially we had a shell of a vaccine facility when the WHO put out a call to host the mRNA hub. And so Afrigen, together with Biovac and the SAMRC, put together this application, and we were successful. And that announcement came out in June of 2021, actually on the 21st of June, 2021. And so that is why our candidate vaccine is called AfriVac, for Afrigen, Africa vaccine, and 2121 for the date. It's been 2 years since that announcement, and 1 year since we actually had the first indication of our vaccine candidate showing immunogenicity in early mouse data. Okay, so let me move on quickly. So you've seen Ike showed how the South African consortium is put together, and Afrigen's role is to be the developer and then to package that technology for transfer to our partners. And we are taking that development to early clinical trial appropriate scale. And we're very well supported by the African Union and the Africa CDC, as well as the Department of Science and Innovation of South Africa, with the WHO and the NPP co-leading the program. Okay, so I know that that you guys are maybe a room full of advocacy people, but I know that everybody is become so technical because we have information at the hand through the internet. And so forgive me if I go a little bit technical, but this is a high-level process flow chart for manufacturing mRNA vaccines. When we started out this program, as Ike mentioned, we didn't have a commercial tech transfer. And so the South African consortium, we needed to develop the technology from scratch. And so we based it off of all the information that was presented in the public domain. And mRNA COVID-19 vaccine was used as this proof of concept product, but with the view to look towards other disease targets that have national and regional needs, and also to look at improvements of the vaccine, as Ike has already mentioned. And Mitchell said so very nicely in the beginning that, you know, all the years of research and development has really built the groundwork for us being able to make mRNA vaccines available for COVID at such a rapid pace. And that came from quite a lot of years based on HIV research, but also on research related to SARS-CoV-1, MERS. And so we had a lot of knowledge about coronaviruses. And so a key aspect of this process is that you have to have access to an antigen. And if you don't have access to an antigen, then you need to design one from the beginning. And that takes a much longer time. And from the antigen, you can design the mRNA cassette. And then from there, you would go on with, you know, the plasmid, which is the starting material for the mRNA synthesis, which is an enzymatic reaction, purification, which is absolutely essential to remove all the impurities that would lead to reactogenicity, formulation for finish, and then after that, you would take your candidate to preclinical studies and then on to clinical studies. And so this picture at the top here just shows you the general structure of the mRNA cassette. And we use public information based off of Spikevax, which is the Moderna vaccine. But there were still very many gaps that we needed to fill. And as we move towards looking at next generation products and addressing these challenges of thermostability, reduced cost of goods, and freedom to operate, we don't want to, we can't do all of this on our own, and we don't want to reinvent things that have already been developed. So we plan, and we are working with many partners through these CRADAs, collaboration, research and development agreements, to get access to specific technologies that accelerate the development process. And I will tell you a little bit more about SAMVAC, which is a South African consortium working on this. And so our main focus at the moment is to demonstrate the platform validity. And we do this, we're going to do this in 2 ways. First, by looking at a collection of all the preclinical data to show comparability of the vaccine candidate Afrivac 2121 with the reference vaccine, which is approved in the market, and then to look at the very strong physicochemical characterization of the vaccine candidates. And then while we do that, to package the manufacturing and the analytical data so that it can be transferred to our partners. And so you've seen this slide just in a different format. I think I actually showed it previously. And what you see here is 15 partners across 4 continents in low middle income countries. And, you know, there was such an eagerness to receive technology and to receive training. And so we were really asked by the WHO to accelerate the program, to start training the folks, even though we hadn't yet fully developed the technology. And so what we did was, as Afrigen and the South African consortium have been building the know-how, we've been packaging it in a way that we can already be transferring to the partners. And so we've trained 11 of the 15 partners already, but this has been on the laboratory scale process. And when we complete the training of the rest of the partners this year, we will follow that on with the actual technology transfer for the process that we developed for the phase 1/2 clinical trial. And so there are 3 key elements that we needed, or that we sort of highlight in terms of this multilateral technology transfer, which is really the biggest of its kind. So I've mentioned that, you know, the strategy for this was largely based off of the success of the tech transfer that happened for the influenza program. But this is a way bigger program with so many partners on board, which really is so exciting, and it will really give us the power to unite as a network of partners, particularly in the case of pandemics. And so there are 3 key elements that we just want to highlight, which Afrigen has been building, which is perfectly aligned with this access model that the WHO and the MPP are driving. And that is the hardware, which is the facilities, which are equipped to enable end-to-end technology development and manufacturing. The software, this is all the systems that we would have in place to ensure quality of the vaccine candidates, and then all the brainware. So this is investment in people, making sure that we have all the integrated skills and the development that's happened. And so in Afrigen, over the last year, we've grown by 300%. And we have amazing scientists and engineers and, you know, pharmacists, veterinarians, and so forth that have joined the team. But there's a large component of that workforce that has never worked in a pharmaceutical industry in a GMP environment. So there's a lot of skills development that is needing to happen. And we're doing this with programs that already are in existence and are being coordinated. So this is basically a picture that shows you the hardware. It's our end-to-end facility where we can do research and development and take it all the way through to manufacturing at a semi-industrial scale, which will be able to make material that can be used in early clinical trials. Okay, so just a little bit on the results. So technical people, scientists, always proceed cautiously when we are giving out results, because we want to be able to show robust, statistically relevant results and that has been peer reviewed. So I just want to say that this is preliminary data, but what it shows you is that when we compare material in a mouse model, which is the Afrivac candidate, in comparison to the reference vaccine, that we have very similar neutralization activity. This is against the original Wuhan variant, and that after the boost, that you have an equal boosting effect at the lower dose and the higher dose. And so this is really great potential in the development of this vaccine. Obviously, we've had a lot of skepticism around this, but this is really evidence that that we can do it, and that so far the candidate is performing well. Currently, we are waiting for the results that from the hamster challenge study, which has actually just finished a day or two ago, and the team focusing very strongly now on process development and scale up. And I just want to acknowledge all the partners that are shown there below that have contributed to this development work. And then I don't have to say much about the value that we've seen of the mRNA vaccines, particularly in COVID, to reduce the severity of illness and to prevent death. But safe to say, and I think most of you will know this, that there were quite a few mRNA vaccines which were in development before the pandemic started. I think about 15 or so that I am aware of. And the development was to look at protection against an array of common pathogens with using disease-specific target strategies. So the slide just gives you a summary of the targets, the challenges that exist, and the strategies that we should be looking for. So if we just look to the HIV side, that we know that we can target conserved regions of the surface glycoproteins, and that what we really want is broadly neutralizing antibodies. But we do face the challenge of rapidly mutating virus, and this is the the big thing that we've been trying to crack for all of these years. I'm not going to go into details of all of this. I'm sure that the slides can be made available to you if you want them. But mRNA technology for vaccines, vaccine development, and to target prophylaxis or even therapeutics is not the silver bullet, and we do need the other vaccine platforms. But it does present unique opportunities, which really includes the rapid way that you can go to manufacture and you can test antigens very quickly. But there are a number of challenges that we need to address, and many of you will know these, and we can perhaps talk about these a bit later. Everybody wants to know where to next after the COVID-19. What are we going to be using mRNA for? We're here to discuss HIV, but just to say that we did an initial assessment which looked at inputs from many different aspects, from key opinion leaders, from all the bigger organizations like Gavi and CEPI, and these are the inputs that we use to help to prioritize the disease targets. So essentially what you would have is a matrix that you would use for assessing or including the mRNA utility. So you would pop in a few of the targets and you would go through all of these things. I won't go into the details, and then you would have some priorities that pop out. And obviously for us, HIV and TB, where we have such a high burden in Africa and in some other low middle income countries, is a priority for us, even though we recognize that it may be a longer term program. And so for the potential for the HIV vaccine, I think that everybody saw this snapshot last year, which shows you the HIV vaccine trials that are ongoing. Some of them are going to come to completion in June, July, and October this year. And so we'll all look to the data for that. But the mRNA platform does allow us to be nimble, to be responsive, be able to screen antigens, and there's definitely potential there. And so we're looking to see what we can do. The South African MRC consortium, which is sort of overseeing the innovation platform, we've already started to look at the development of an HIV subtype C mRNA vaccine, which expresses ENV and the envelope protein and GAG antigens. And we've also applied for this HIV VISTA program, which is being funded by the USAID. And, you know, it's leveraging off of many years of research that has happened here in South Africa, a lot in the clinical trial space. And so using those inputs into the vaccine design space. And so this is my last slide, and it just shows you the local innovation platform, which has been driven by the South African mRNA Vaccine Consortium. And what we seek to do is to address the full value chain in terms of vaccine development with partners all across South Africa and even some in Africa. And we're looking at everything from genomics all the way through to clinical trials and commercial manufacturing. And our targets at the moment for this particular program are TB and HIV, and some other regional priorities. And then lastly, just to thank everybody, as they say, it takes a village. And so we have really been so well supported by so many in this program, and we are very grateful. And thank you all for your attention. I'll hand over back to Mitchell.
Caryn, thank you so much. That was fantastic to see this come alive over just the last couple of years. And really congratulations and thank you. I'm sure I know there are lots of questions, and we'll get to those in a couple of minutes, but I first want to turn to another dear friend and colleague, Mike Frick from the Treatment Action Group. And I think everybody knows TAG and AVAC have partnered longer than I think any of us care to count, from both organizations' beginnings almost 30 years ago. And Mike co-directs the TB program at TAG. And you may ask, why a TB advocate view? And I think one of the things that Stacey, Hannah, and I and others at AVAC have seen in our collaborations with Mike and with the whole TAG team is how clearly related these issues are and how we are stronger together. As Caryn just said, it takes a village, it takes advocates of all types. And Mike's perspective on the TB pipeline of vaccine development, but also on these larger sets of issues are exactly the right thing for this audience. So Mike, over to you.
All right. Thank you, Mitchell. Hi, good morning to everybody from here in New York. And big thank you to the AVAC team for bringing us together today. I was asked to give a TB advocate's perspective on mRNA vaccines for TB and HIV in local manufacturing. And like Mitchell said, I feel very honored to be speaking as a TB advocate on a call to mark HIV Vaccine Awareness Day. So thank you for including me in this field and conversation. And with the mRNA Hub partners on the call with me, Caryn and Ike, I did want to open with this photo from Cape Town in 2015, when TB activists demonstrated in front of then Health Minister Aaron Motsoaledi. And the central ask of the demonstration was to invest in TB research. And with the hub's location at Afrigen, BioVac, and the Medical Research Council in Cape Town, I think that call has at least partly been answered, especially given their early work on TB and HIV vaccines. I wanted to start with a bit of history because I think a lot of the current conversation around technology transfer for vaccines assumes that calls for tech transfer are calls to move knowledge from the north to the south, when really originally vaccination as a technology was a south to north transfer, and one that was often made under very unequal conditions. So hundreds of years before Jenner introduced variolation against smallpox in England with the help of Montague and others, China, India, the Ottoman Empire were using variolation as a technique against smallpox. Vaccination against smallpox in America actually arrived via the slave trade. Cotton Mather, the sort of firebrand Puritan preacher of New England colony, is widely credited with introducing vaccination against smallpox to the colony. But he learned about the technique, he was a slave owner, from one of his slaves, a man named Onesmus, who was born in what is today Libya. And as Onesmus explained to him, variolation vaccination against smallpox was something common and done often where he was from. And it was that technique that Cotton Mather introduced into New England. And so I think it's just important for those of us on the line who are in the global north, for us and for our governments, to acknowledge the fact that the basic techniques of vaccination did not originate in our societies, that they were shared with us and often taken by us. And I think it's important to foreground the kind of conversations we're having today about technology transfer in this history and not to forget where we started. I think we also need to remember, as Mitchell has pointed out already, and Caryn as well, that mRNA did not start with the pandemic. That work on mRNA and its story began well before the pandemic started, and it really started with HIV and other things. But it's also a story for TB that began before the pandemic. So in 2019, months before we knew, the world that we were about to enter with COVID-19, BioNTech and the Gates Foundation announced a collaboration to develop mRNA vaccines for TB and HIV based on an initial equity investment from Gates, with the potential for that to nearly double for clinical evaluation work down the line. And this press release ended with, assurances of affordable access to the candidates that would come out of this work in developing countries, with BioNTech maintaining rights to commercialize them in the developed world. And I think what COVID taught us is to ask what specifically these types of assurances mean, and to ask a bigger question about, is there a better way to develop vaccines against these diseases? Can we locate manufacturing and research in the global south to begin with? And so today the field is very crowded, and the mRNA vaccines are coming for TB. This slide summarizes the players who are actively developing TB vaccines based on mRNA. That BioNTech work has progressed to the point where they have investigational mRNA vaccine candidate under the common name BNT164, with 2 trials registered, one in Germany and the other in South Africa. IAVI and Moderna have announced their collaboration to develop mRNA vaccines against a range of diseases, including HIV and TB, with the TB vaccine constructs undergoing preclinical evaluation. And then the mRNA Tech Transfer Hub has started discovering preclinical work on TB vaccine candidates. And TB was discussed at a day-long meeting at the Hub's week-long convening in Cape Town in April 2023, which I was lucky enough to attend. And there are other players who are entering the space as well. Small biotech companies such as PopVax in India are also entering this domain. This is the TB vaccine pipeline, which I show mainly just to point out the different candidates that we have. It's still largely a pipeline, not of mRNA-based candidates, but of more traditional protein adjuvant, viral vectored, or whole cell mycobacterial vaccine candidates. And for the HIV advocates on the line, you can see that at the moment, the TB vaccine pipeline is very top heavy. We're really weighted toward phase 3 trials. And a big focus has been the excitement around initiating and completing several phase 3 studies of different TB vaccines. But at the same time, we're acutely aware that phase 1 looks pretty empty for us. And we need to populate our pipeline with new candidates and new constructs. And in blue on this slide is that first mRNA TB vaccine, which entered phase 1 clinical trials through BioNTech. And that first trial in Germany enrolled its first participant last month. So we're off to the races, and we'll see where that takes us. We also know that the mRNA vaccines are coming for HIV. So this is the AVAC brief that Caryn showed as well, providing a snapshot of 3 phase 1 clinical trials using mRNA platform for HIV vaccine development and discovery. I think it's interesting to note that the commercial partner for all 3 of these efforts is Moderna. And when I was at the Hub meeting in April, a senior HIV vaccine researcher made the following comment and sort of summarizing mRNA and its applicability for HIV. I'm not quoting them by name because it's a paraphrase here from my notes, which I took by hand and had trouble reading after the fact. So I didn't want to misquote, but the gist of it was that mRNA is a necessary tool to iterate to create the vaccine regimen we think will be necessary to coax immunity toward what we think is likely to work. So there are some qualifiers there, and I qualified the previous slides by putting that maybe into parentheses, and I think it's worth unpacking why I did that. I think there's really nothing inherently magic about mRNA. The AVAC brief describes it, very directly as a platform to deliver a piece of genetic material that instructs the body to make a protein fragment of the target pathogen, which the immune system will then recognize and react to. But for that delivery to happen, it's helpful to know what should be delivered, or what's the right antigen target, what does protective immunity require, and how best to use vaccines to achieve it. And for HIV and TB, this journey, even with the use of mRNA technology, is not going to be a 100-day dash to the finish line of a vaccine for us in our communities. We are running a different type of race. We have lots of hurdles and obstacles that we're going to have to jump over before we get to the finish line. And mRNA isn't going to remove those obstacles, but it may help us get over them more quickly and with more style. And so again, it's not going to be easy for HIV and TB, and the reasons I think are well appreciated by this group. For HIV, they've already been mentioned by the previous speakers, the kind of astonishing genetic diversity of the HIV virus, its rapid mutations, its complex mechanisms of immune evasion, the fact that it actually targets and integrates into the very immune cells that we rely on for so much of our immunity. TB has its own challenges. TB has co-evolved with humanity for tens of thousands of years. It's learned all the tricks of our immune system. It has a latent character, a fourth of humanity is latently infected with TB infection. And TB, unlike a virus, is a bacteria, so it has a very large genome, 4,000 protein coding genes. Difficult to know which of those need to be targeted by a vaccine, and which are best recognized by the immune response that we need to create to generate protective immunity against either TB infection or TB disease. So really complex challenges for both of these pathogens. I really liked what Petro Terblanche said at the Hub meeting in April, that we are not building a single product, we are building a platform. And I think it's this platform characteristic of mRNA that makes it so promising. So mRNA itself is no panacea, but it is a tool, not the only one, but an important one that we can deploy as part of a larger strategy to develop vaccines. This tool allows us to iterate early, and in a sense, I think de-risks some of the fear of failure by allowing us to fail faster, earlier, and at less cost, to try different combinations of antigens, go after different targets, see what works in preclinical or early clinical development, iterate on that, start over if we need to, and to do that more quickly than we're able to do with other vaccine platforms. And mRNA is a platform technology on which many applications can be built, but only if it's shared and open to many different developers. And I think that's why the work of the Hub is so exciting. And as has been pointed out, this platform is still evolving and fairly young, and there are lots of improvements to be made to it in terms of cost of goods, in terms of the reactogenicity of the mRNA vaccines for COVID, thermostability, and other factors. And these improvements will happen faster if many different people and groups are working on them. So mRNA doesn't remove the obstacle on our race, but it may be a kind of springboard or platform that helps us to jump over it with greater distance. I also want to point out that vaccines and vaccine platforms, whether mRNA or anything else, don't exist in a vacuum. And so the work that we're doing on mRNA for these diseases sits alongside the many other ways that we can prevent HIV and TB. I know that in HIV, you have a choice agenda for HIV prevention. We have something similar in tuberculosis. You know, TB can be prevented through infection control, through preventive treatment, which is sort of our PrEP equivalent, short course regimens against TB that can be taken in as little as 28 days or 12 weeks. We do have a TB vaccine that protects infants and young children. We're hoping to get new ones to protect adolescents and adults. And then for anyone living with HIV at risk of TB, ART has a protective effect, especially if paired with TB preventive treatment. And all of this depends on social protection, respect for human rights, poverty elimination, and the social determinants of health, like housing, decarceration, and all those things that impact our epidemics. So I think that we have to acknowledge that these vaccines are being developed against this larger background and alongside these other technologies, and we can't be working in isolation. And that makes community engagement and mRNA vaccine development so critical. So with Hub partners on the line today, I do want to take the opportunity as an advocate to say, and I said this in Cape Town, I really think the Hub needs to develop an intentional program of community engagement that engages community and civil society in the diversity of its skill set. It should be a program that gives community a voice in shaping vaccine design. There are many advocates with incredible technical capacities that we've developed, over the years of advocating for the vaccines that we need. We can have community advisory boards, inform study protocols, ensure trials are inclusive of the populations most at risk. We need to shape the acceptability of what we're working on, work to address misinformation about mRNA, create demand, and then many in civil society can help overcome the IP minefield and navigate that freedom to operate that's so important. So manifold benefits to engaging communities in this work. And I really hope the Hub takes this and develops it as an intentional, explicit part of the program it's creating. I want to end by quoting Glenda Gray of the South African Medical Research Council. This is actually a throwback to something that she said in 2016 at the AIDS conference in Durban. There was a meeting on TB and HIV vaccine development. And she said at that meeting that we need good science like we need clean water. I wrote this down in clear handwriting, and I've been quoting it ever since. And I like this quote because to me, it expresses what is really at the heart of the work we do at Treatment Action Group, which is to ensure that everybody can enjoy the fundamental human right to enjoy the benefits of scientific progress and its applications without discrimination. Science, like water, is a human right, and we need it. And under the right to science, access, if we think about equitable access, is defined in terms of 3 dimensions that governments have to work on simultaneously. They need to develop good science. That means investing in research and channeling those investments in a purposive development to solve the problems and challenges that marginalized and disadvantaged groups face preferentially. They need to diffuse the benefits of that science to ensure access to it, non-discrimination and participation in the science, and they need to conserve it to ensure that it's available not just for people today, but for people in the future, not just for a privileged few, but for the many. And I think for me as a human rights advocate and as a science-based advocate, what makes me so excited about the Hub program is the fact that it's one of the few development programs that is working on development, diffusion, conservation at the same time as part of what I call that R&D and manufacturing integrated network. So it's an integrated network of R&D and manufacturing. I think it's also an integrated network of right to science concepts in terms of these 3 tripartite obligations that we hold so central. So with that, I will stop and just say thank you, and really looking forward to the discussion and to seeing what comes out of the work that the Hub is doing. Thanks very much.
Great, Mike, that was brilliant. Mike, Caryn, and Ike, all 3 of you, phenomenal presentations. I know that we are near the top of the hour. I do hope people can stay longer because there is so much to dig into, and want to invite people to certainly Mike, Caryn, and Ike to join on screen, and want to invite people to put questions and comments into the chat or the Q&A. We'll take them wherever they come. And also, if you want to raise your hand, we'll get you off mute. We'd love to take questions. That was just brilliant throughout. I wonder, a couple of questions did come in as you all were going, and maybe I'll start with those. And one of the fundamental principles, and it was beautiful how you ended it, Mike, in terms of what we think of at AVAC as the R&D&D, the research and development and delivery framework, and including manufacturing and access. Clearly, intellectual property matters. Jonas Salk said, when asked why he didn't patent the polio vaccine, he said, can you patent the sun? Clearly, that is a very different experience than what we saw the last 3 years. But I wonder, particularly Mike and Caryn, or Ike and Caryn, you all talked about the Hub and the process of both WHO and MPP's involvement. and now the hub taking it forward. Where does, particularly for the newer products as they get developed coming through the hub, where will intellectual property sit? How does the licensing work for things that come out of the hub, given the really elegant but complex kind of network map that both Ike and Caryn showed? So I don't know, Ike or Caryn, if one of you want to start with that.
I think I'll defer to Ike.
Hi, Mitchell, really good question. So with the selected manufacturing partners, we have technology transfer agreements where the framework has been built that the technology that is transferred to them, they have the opportunity to improve this, not only in terms of the technology itself, but in disease areas as well. Mm. And with that improvement, the framework looks at them sharing this technology back within the network, royalty-free, that will be made available to all of the manufacturing partners to not only manufacture that product or using that technology, but further improve it themselves. So it's generally, the framework is based on a manufacturing and R&D network where all of the IP is shared.
Great, super. Another question, and Mike, you described it. You know, mRNA is potentially a great tool for any number of pathogens, but not the only tool. And I wonder if any of the three of you, you know, we talk about the mRNA hub, but so much of what you each described, one could imagine looking at other platforms. And I know, Caryn, you had a great slide looking at the advantages and challenges of mRNA itself, but is there already conversations about hubs looking at other platform technologies, or is there a sense that given that kind of plug and play nature that you described, Caryn, that mRNA is the best hope for a hub and spoke approach? But or are there other potential platforms? And is the mRNA hub looking at those other platforms, I guess would be the secondary question. And that's open to all of you.
Okay, let me take that one first, and then I'll hand over to Ike and Mike. So I think Ike mentioned very early on in his talk that, you know, it was a strategic decision to go with mRNA first, but the longer term view is to replicate this kind of model for other platforms. And there's already been discussions of what these will look like subunit proteins and the adenoviral vector-based platforms. And you know, I think that each of these platforms have their specific role to play, and we have to, it's very complicated, right? So it depends on a lot of things, like how much you can sell that particular vaccine for, and a particular technology will lend itself more towards a lower cost, if I can put it that way. And so I really want to just emphasize that even though we've been working on mRNA, that there's value for every single other platform. And I think that the intention is that now that we have this framework and we're starting to see how everything needs to work together, that kind of framework can be applied to the other technologies. So I'll stop there.
Great.
Maybe add, you know, from my perspective, I think the other platform technology in the vaccine space is to think of an adjuvant or an adjuvant system as a type of platform that can be used across different vaccines. And that has already become very relevant in TB vaccine development. The sort of leading candidate ready for phase 3 trials is called M72 AS01. It's the AS01 that's the adjuvant owned by GlaxoSmithKline, used by them in this TB vaccine, in the RTSS vaccine for malaria, in the Shingrix vaccine, in the new RSV vaccine they just had approved. And the molecule in that's so central, QS21, is used by Novavax in their Matrix M adjuvant construct. So I would say, you know, if you're interested in platform technologies for vaccines, look at QS21, think of adjuvant systems, very proprietary. You know, we don't have programs like the hub for adjuvancy yet, but that's coming, and I know efforts are underway to do adjuvant work. But I think that's an area where I would say we need advocate attention in terms of freedom to operate and access to technologies, not just for end users, but for scientists working on different diseases.
Mike, that's a great point. I was actually so happy, Caryn, about your own background in adjuvants, because I think Mike's exactly right. And just, I don't know if anyone's still on the line from the military HIV research program, but just to flag, although not a hub itself, the MHRP has developed and licensed and is making widely available to anyone an adjuvant that grew out of the MHRP program, really with that in mind, not to be beholden to the IP of an adjuvant, because it is really a platform. So super, super helpful. There's a Roger Tatu, a longtime advocate and partner, has a great question in the chat that I'd love to have all of you talk about. We get all excited and sometimes geek out on mRNA or on adjuvants, but some of these newer technologies do foster, particularly in the current environment, misinformation. And would love to hear people, and I think it relates, Mike, to your point about the essential need of community engagement. But as vaccines get developed and rolled out, some of the newer technologies have been hot buttons for conspiracy theory and misinformation and active disinformation. And I'm wondering if, Caryn, if you're experiencing any of that within the mRNA hub as people just talk about it, but also to all three of you, how might we begin to address that in the development space as much as in the manufacturing and delivery spaces?
Yeah, so I think that, we should go back to Mike's last slide, because I think that is like really where the crux of the matter is. So for us as the vaccine manufacturer, we hear this thing of like vaccine hesitancy being thrown out all the time. And we feel like we really need the social scientists. We need to work together to understand it, because like I said in my talk, like people have access to information. And like you said, Mitchell, misinformation. But we can't just assume ignorance in many things. And I think sometimes that's one of the things, because we say we need to educate. And I think that there needs to really be such a strong collaboration between us, the developers, and the social scientists and advocacy groups to really understand what contributes to hesitancy. I probably know less of this than you all do. And then we can really start to address it. We've definitely experienced it. And, the mRNA story about, the fear of integration with your own DNA and all of these kinds of things. So we need the scientists and we need the healthcare practitioners to really be, helping in that space. And yeah, I think it's a real team effort. Yeah. Great.
I might just, maybe this is something to continue post to this call, but I think building on Mike's slide, but Caryn, also your kind of, and Ike too, your organograms of the hub and perhaps adding both a deep and durable community engagement orb within the overall hub and a social and behavioral science hub or orb within the structure. I think both of those would be great additions, just because these become issues often much later in the process, too late, rather than, I think Jim Pickett in the chat talks about pre-bunking, doing this in advance and bringing the social, behavioral, and community perspectives in. So just a commentary there. I know we're over, but people are still here, and that's great. A great question from Ali Bancroft from Health Gap in the chat about how we might build political will. And obviously WHO at a global level and MPP really bought into this concept, drove this concept. Ike, how might we, how is political commitment? The map you showed has lots of countries. Are we seeing political leaders in those countries get as excited as the researchers and the manufacturers? And if not, what do we need to do about that? Oh, Ike, you're on mute though.
Apologies. Excellent question. So the map that I showed earlier is really looked at the countries where the member states had put in an expression of interest for a selection of manufacturing partners that were positioned in their country. So this was their ask, and basically everyone that applied, they were asked to select their manufacturer, their local manufacturer. On top of that, the WHO are really understand that political will is a key enabler to this. So they have a series of networking with each of the governments, ensuring that there's alignment with the ministers of health, ministers of science, trade and industry, so that when the manufacturing partner comes up with a strategy, it's a strategy that is bought in by the country and the region.
Great. And I think, Caryn, you talked about the African Union and the African Centers for Disease Control and Prevention and their support for the hub. Is there more you can say? I mean, their logos look great on the slide. How are the AU and the Africa CDC collaborating with you?
Yeah, so that I think there's just been a new director of the Africa CDC just a week or so ago, right? So just to show the support, he was in Afrigen like that same week. And we were able to welcome him. And so I think what is difficult is that with the pandemic, there was urgency and there was political will. And then as sort of the pandemic seems to fade a little bit, one of the key things that MPP and WHO with us have been trying to do is to keep everybody engaged. And what that means, you've got to keep momentum. And so that's a key thing for the program. And also Afrigen as a hub, as much as it has been quite onerous on us, what we would call sort of the additional things, we have really been open to have people come in and see. And this includes our political leaders. So, we even had the King and Queen of Belgium come in and be able to talk to them about the technology. And so I think it's just keeping that engagement and showing that there's movement and that this whole hub program has not just been talk. It's like we are in action mode and we're going to do what we said we're going to do. So I think that's key.
No, that's great. And I will just say, and I'll put in the chat, in December, the previous and founding director of the Africa CDC, who of course is now the ambassador leading the US PEPFAR program, talked about the critical importance of bold targets in the HIV space for African manufacturing. Of course, that's in the short term, looking at antiretroviral treatment, perhaps hopefully soon African manufactured injectable PrEP, but perhaps also a signal of building a platform. And given Ambassador Nkengasong's early work in HIV vaccines, I hope a commitment to HIV and TB vaccine research and development and manufacturing on the continent. A question came in and I do want to come back to about Moderna and BioNTech. I think all three of you at different points mentioned those two companies, amongst others. Caryn, you mentioned that you had to rely on what was in the public domain to be able to create things, even though a vaccine had two vaccines using mRNA had already been created. What is the relationship between the hubs and Moderna, BioNTech, and Pfizer? And are there ways to go faster together? And how competitive does it become with the hub working in a number of African countries, and now Moderna talking about a Kenyan vaccine manufacturing site, BioNTech flying in containers to Rwanda, et cetera. So what is the relationship with the innovators, the developers, and are there ways to build collaborations, or is it just a separate and parallel universe?
So I'm going to give Ike the opportunity to speak first, if he would like to.
Yeah, I think, Caryn, I think that you can talk on the ongoing discussions that you've had, that Afrigen have had with Moderna. But just to say that the Moderna technology, the BioNTech technology, this was established and product was made available during the pandemic. Of course, if this technology was made available and transferred to Afrigen, we may be further along the line now. This didn't happen, and so we were forced to develop our own mRNA platform. We have used this first generation platform for a few reasons. Firstly, to demonstrate a proof of concept. However, we understand that this first generation technology is only the beginning, and that we really need to be looking at the next generation technologies, because we foresee that there's going to be there's significant improvement opportunities. And that's where this creation of this R&D network is really going to be of value, where we'll be generating technologies that we won't have freedom to operate issues. But as you said, BioNTech and Moderna, they are coming to Africa. And in terms of looking at, addressing the issues in terms of providing access to life-saving medicines in Africa, I mean, we obviously welcome that. There is a contribution. It's not aligned with our model. And so we don't see a place for that in our model at this stage. And so, yeah, we're happy to go on with our business on that. Let me pause there and then maybe let Caryn contribute as well.
Yeah, so what I can say is it's usually complicated, as I am sure you can all imagine. There are many layers in this discussion, but what we've tried to do is, because we are collaborative, we can't do this on our own, and we still also believe that the global north and the global south and the high income countries and the low income countries still need to collaborate. That is just a given. There's a lot that still needs to be learned, and there are things that need to be transferred and shared. But the narrative that we want to bring and that we want to change is that we're not only the consumers of IP, we are the generators of IP. As Mike took us back, to the smallpox. And so I think that where we are positioned now is really attractive for collaboration, even for the likes of Pfizer and Moderna. And we are open. We are still open. There are different ways to collaborate. We have MPP there to help us navigate the space, where it's complicated with IP and so forth. But we certainly do feel that wherever there's a way that we can move things faster, we want to do that. And we don't want the management of IP to be a barrier. And I think in the past it has been, but we have to manage it carefully. And we also have to think, and I know that in certain spaces that, we don't want to talk about, the value for shareholders and all of that, but that's a true thing. And the other important point is that being in the biotechnology space, we always want to incentivize innovation. And so this approach to sort of voluntary licensing and so forth. So we've had, we've been caught in the middle with that as well. And we've wanted to say that we would love to collaborate, and we would love to have a voluntary license, and we want to keep on incentivizing innovation. Yeah, so I'm going to stop there. Mike has his hand up.
Yeah, no, brilliantly said, Caryn. I was going to turn to you, Mike. Hand or not?
I raised it just for dramatic effect. I can't help but point out that I think the thing that BioNTech, Moderna, and the Hub all have in common is that behind them is governments doing a lot of the basic research that is then turned into products. And so just as the Hub, most of the funders are governments in the global north at the moment, Canada and European countries. So too, the Moderna vaccine was certainly an NIH vaccine, and BioNTech and Pfizer got a lot of help from governments and from entities whose work is made possible by governments, philanthropies, which are benefiting from tax systems that governments establish to allow them to work. So I think there's actually a lot at common at the root. And to the question about political will, I think that we can think of governments investing in the Hub's work directly, and that needs to happen, through funding. But there's also the ability of governments to license their technology that they own to multiple partners, including the Hub. And so contributions and will can also be demonstrated in kind. And I think we need to look at the licensing arrangements as a demonstration of political will, in addition just to kind of the funding that's announced. And as Ike said, it's about a coherence between health, industrial, and trade policy. And we know at the moment it's incoherent. In 2018, right before the pandemic, the UN Secretary General published a report on reconciling the incoherence of health and intellectual property and trade law. And I think that report, has a lot of lessons that are still very relevant today. And if we had acted on it then, we would have been in a very different situation with the pandemic when it happened several years later. just a few reflections on my side. Thanks.
Oh, brilliant reflections, Mike. And you know, it's so important because as hard as the science is, whether it's genomic sequencing or vaccine manufacturing and everything in between, the political issues you just described shouldn't be harder than the science, but inevitably they are. I did just want very quickly, and I'm going to invite anybody to raise their hand if people want to come off mute and be heard. But just you know, it's hard not to, you know, this is in keeping with our HIV Vaccine Awareness Day, which just was May 18th. And while we don't have an HIV vaccine, as everyone knows, one of the most important outcomes of President Clinton's speech in May 1997 was the establishment of the Vaccine Research Center that you just mentioned, Mike. And without the VRC, we would not have, I would argue, as quickly at least, or at all, at least the Moderna, called the Moderna vaccine, but really the NIH Moderna vaccine. That same team that led to that vaccine development also was the team behind one of the RSV vaccines that was just licensed a few months ago by the FDA. It is a brilliant example of political will, of establishing the NIH's Vaccine Research Center that has delivered a COVID vaccine and an RSV vaccine, both of which did go in to be licensed by large pharmaceutical companies and sold at quite high prices. And to the point in the chat from my colleague Annette, public pay has not resulted in public say in the way that it should. But I think to your point, Mike, this is an opportunity to reimagine some of these global constructs in terms of how this happens. A mini editorial for me on Vaccine Awareness Day. I'm going to pause just to see if anybody wants to put a hand up in the chat, in the participant list, and ask a question live. All right, I paused, I didn't see them. What I'd love is in our last couple of minutes, maybe to give each of the three of you just a couple of minutes to reflect on, you know, we obviously want a TB vaccine, an additional TB vaccine, safe and effective and affordable and accessible. We obviously want an HIV vaccine that is safe and effective and accessible and affordable. That's not going to happen in the next year. What does success look like when we think about the platforms, the hub, over the next, say, 12 to 18 months? What are you going to look for, say, by the end of 2024, that would give you a sense that we are in a better place scientifically and politically with respect to vaccine development? Picking up on, Mike, your last point. So maybe I'll go in reverse order and start with you. What does success look like a year from now that we as advocates can hold ourselves, each other, and other stakeholders to account?
I think for TB, we really need to get into clinic. You know, we've had some candidates that need to start late-stage efficacy trials, and those have been delayed. You know, we had a positive efficacy signal for one of them in 2018, and here we are in 2023, and the trial has not yet opened. So that's a gap that we can't repeat again, and we need to just get that study underway. And that's important for the work of the hub, because those late-stage efficacy trials are going to generate the samples, the immunogenicity data that we need to inform the early vaccine work that can then be used to kind of plug and play into the mRNA platform. And so I also want to see a commitment to like rapid sample sharing, to data from those trials becoming made available as soon as it's you know, at our fingertips, and we can do so based on, you know, trial ethics and so forth. And just have a really much more collaboration on the discovery space linked to the clinical work that we're doing on some of these legacy constructs, which may end up working, but even if they don't, you know, represent, I think, an enormously important investment in the next generation of vaccines that we need to be developing.
That's great. Share the samples and close the gap. You know, COVID was able to run concurrent trials. 2B trials were folded into phase 3, and both in TB and HIV, we get a positive phase 2B result, and then we wait 6 years to try to see if there's money in manufacturing to even start a phase 3. We've got to close the gap. Caryn, over the next year, what does success look like for you in the hub?
Yeah, so there's a lot of things. I will choose two things to say. The first one will be that, you know, as Mike so eloquently pointed out, that we need pipeline to feed into phase 1, right? So what we've seen is that there's quite a gap in terms of funders for the earlier stuff. So you can't get to the phase 1 if you don't have the earlier stuff being funded. So we are really working with the funders on this, and NPP and WHO supporting us in this. So there's early work that needs to be done, and it needs to be funded well. That's the one thing. The second thing I want to say is that one of the priorities for the hub is to look at, reducing the cost of the goods for the mRNA vaccines. And so we have been evaluating some technologies. And so in a year from now, we want to show data on the hypotheses for these next generation technologies that give sort of contextual costs of goods. So not what they cost to be made in high income countries. What do they really cost to be made here where we are?
Brilliant. That is so great, Caryn. Thank you. Ike.
Mitchell, for me, the mRNA technology transfer program is unique. The model is unique. And for me, demonstrating that this could be used for other platforms would be the ultimate goal. Obviously, there's several steps to be able to demonstrate success of this, and giving the confidence that this type of model could be used in other platforms. But for me, ultimately, this would be the cherry on top, demonstrating that this could be used for other models, and seeing a manufacturing network. Often manufacturers work in silos, but this network concept that is fully supported by R&D, really looking at creating this future pandemic integrated with interpandemic sustainability model. For me, that would be the cherry on top.
I love it. All three of you, brilliant summaries. It reminds one of the smartest, wisest HIV vaccine advocates, Bill Snow, once said, some things just take as long as they take, and we can't change that. But for the things that we can, let's identify them and squeeze that time out of them. And what each of you said, whether that's closing the gap, investing in the pipeline, reducing the COGS of the platform, Caryn, proving the concept of these models, those things can be done in a year's time. Doesn't mean we get a vaccine out the door in that year, but if we can do each of the things you all named, the field of vaccinology, HIV, TB, and frankly, broadly for vaccines, will be a better place. And so thank you all for identifying those. One of the things about HIV Vaccine Awareness Day is to try to be reminded that there is hope and optimism in the midst of challenging science and of, as we said at the beginning of the call, of insane politics in the case of Uganda. And it's important to keep hope and expectation and excitement alive. And I think you three have done that brilliantly. I do want to encourage everybody to tune in a week from tomorrow. Or no, a week from yesterday. Next, I want to make sure I get the date right. Next Wednesday, June 7th, we will do a second webinar, same time, focused on antibodies. And many of you will know that there's been a proliferation of antibody-focused research. Caryn described the hunt for a neutralizing antibody approach for vaccines, but antibodies being also developed as products of their own. We have an amazing panel joining us next week. It's Wednesday, June 7th. I think someone will put the link in the chat. It'll be on the AVAC website, and want to encourage people to join there. Thank you so much, Levi. Again, Caryn, Ike, and Mike, this was brilliant. I want to thank everybody for sticking with us. We will be posting the slides and the recording for those of you who came late, or if people are already off and they need to hear what they missed. And let's keep the conversation going. And we look forward to working with all of you on the way forward. Have great evenings in Switzerland and South Africa. And great afternoons in elsewhere. But thank you all, and we'll talk soon.
Thanks very much.
Thank you for having us.
Cheers all. Thank you.